Go. Hello, welcome to biochemistry. Boy, it seems like we're flying through this. Okay, biochemistry. Biochemistry is the study of molecules and their reactions in living organisms. Okay, so what makes this cooler? I mean different. No, I don't. I really mean cooler. What makes this more fun is that we are talking about chemistry in living organisms. It's like biology and chemistry put together. It's like, okay, we have all this chemicals and all this stuff. Who cares? Oh, I care because it happens in my body. So that's what biochemistry is. It's like you have these cycles, you have this physiology, you have this thing going on. What is making it do that? And what's making it do that is chemistry, really. It's the way the bonds connect to each other. It's how they connect. It's how they break apart. It's all that good stuff. So that is biochemistry, the study of molecules. So health science really is biochemistry. Physiology is really biochemistry. We just call it something else so people aren't terrified of it. Okay. Physicians are faced with biochemistry every day. Diseases, abnormalities, all these things are related to biochemistry. The goal of biochemistry is to understand the structures and the biomolecules going on in your body and their relationship to the structure and function. So underline this, make it red, do something awesome to it. Structure and function. So basically, structure and function go together. Function and structure are the same. So you look at something, you're like, that's so weird. Why does it do that? If you really, really pay attention to the chemistry, you can figure it out because the function, what it does, goes along with the structure. What it looks like, how it reacts, all that stuff. Function and structure go together. This is important. Biomolecules. They vary in size. You have little tiny, like 1 carbon deals all the way up to bazillion chain polypeptide things that go on forever. But each molecule is made up of carbon. So that's why we have to study organic carbon. It has a carbon skeleton. And then it has all the super fun functional groups. Remember all the functional groups that you learned? Why can I not spell functional groups? I do not know. Let's make it red anyway. Functional groups. Remember, you're learning them now. Yay, functional groups. Carboxylic acids and all that good stuff. So it's the functional groups that are actually interacting with one another to come up with what's going on with you. So that's where you get the functional groups. The major class of biomolecules are proteins, carbohydrates, lipids, and nucleic acid. Each one of these has their own chapter. Yeah, so we'll get to that. Don't worry. Very exciting. So here's a review. Put it in your notes again. The more I see it, the more it just goes in automatically. Carboxylic acid and all those things. Those things are going to interact with each other to form different proteins. They're going to do different things and make up your body. That's going to be awesome. Okay, let's talk about protein structure and function. Proteins are polymers of, who knows the answer to this? Amino acids. Basically, if you took any biology in high school, you talked about amino acids and proteins. But this should be a review for most of you. It might be brand new, like life-changing information from others. It's fun for me either way. Okay, amino acids contain. What do amino acids contain? These should all sound super familiar. And amine. Why amine? I put it there just in case you forgot, which I know you did not. An amine group. So they all contain an amine group. They also contain a carboxylic acid, our carboxyl group. And they also contain the super fun thing that I like to call a side chain. Make it red. Side chain. Not with an E. Okay, a side chain. So every amino acid has an amine group, a carboxyl group, and a side chain. All of them. Next test, I'll make you learn the 20 amino acids. Yay! But for now, you need to know the basic structure of an amino acid, which is written here. An amino acid. That it has an amine group, a carboxylic acid, and a side chain. That's what you need to know. Until the next test, when you actually need to memorize all the amino acids. Now you're memorizing functional groups, so I'm not making you memorize the amino acids. Okay. Shall we talk about amino acids? Yes, we shall. 2. Now you should be able to get this without me telling you, but I'm going to tell you anyway. 2 amino acids form a dipeptide. Okay, who wants to guess what 3 amino acids form? Come on, try. I know you can do it. I know you can do it because you're so smart. Tripeptide. Many amino acids together form a, come on, give it to me, polypeptide. So if it's a polypeptide and I say, does it have 2 or 3 or more? It has more because it's a poly. It doesn't have 2. It doesn't have 3. It has more than that. It can have anywhere from 4 to a bazillion, but it doesn't have 2 or 3. Okay. So you can have 2 amino acids, 3 amino acids, a dipeptide, a tripeptide, or a polypeptide, which is usually like a gigantic bunch of stuff. Let's talk about structure. There are 4 structures. What am I talking about? What does this mean? Primary structure. This is, I call it beads. That's beans, not beans. Beads on a chain. Although beans on a chain would work if you can get them on a chain. Beads on a chain is what I call them. Basically, it is a sequence of amino acids in a protein. Wow, it's like I'm not even trying to spell. Protein chain. Okay, so if I could draw it here, I would draw it like a 0 and a, oh no, I would draw it like a 0 and a dot and a 0 and a dot, like that. Basically, it's like this. Beads on a chain. Well, and do this like this. Okay. It's like beads on a chain. Each one of these is a different amino acid. Beads on a chain. So this is guanine, leucine, isoleucine, like this. How about this one? This is more fun because everyone's a different color. Each one of these different colors represents an amino acid. Primary structure. Now, what goes wrong with this? You have a DNA makes your amino acid. We'll talk about it in 25. Just trust me. DNA makes up your amino acid chain. Amino acid chain, if one of these, so you see this red one here? Do you see that? It's red. Let's say there's a DNA that's wrong. So now I take that red one and it's blue, and it's going to mess stuff up. But this is the primary chain. We're going to see how it could potentially mess things up in a minute. This is the primary chain. Secondary structure. Secondary structure is, let's move this up so we can all get on the same, is the, how do I have that? Regular and repeated spatial organization of neighboring segments of a single protein chain. So we still just have one, right? We still have one necklace or one whatever. Boom. Now, what is this? This is a big long piece of stuff. What's it gonna do? It's gonna form like this. It's gonna form around itself. Let's put it on my pen like this. It's gonna form either an alpha helix. This would be an alpha helix because it's a helix. See that? Isn't that beautiful? How's it going to do that? What's it going to do? Actually, it's going to scrunch up like this a little bit more. It's going to interact with itself. Some of these are hydrogen bonds. Some of these are disulfide bridges. Some of these are London dispersion forces. And it's going to interact with itself. It's going to do this really awesome because it's got all kinds of side chains coming off of it. So let's go back to my red one. Can we see it there? See my red one? I think it's right here. This red one. Do you see it? This red one. Remember, we messed with that. We have a DNA malfunction. We messed with it. And now it's like another color. It's supposed to be red. It's not. Well, if we don't do it, it may not line up right. Let's say it doesn't line up right and it does this. Now we're in trouble, right? This is why one DNA mutation messes with primary, secondary, and tertiary structure. Another one that happens is a beta pleated sheet, but we'll get to that. Don't worry. So now we have tertiary structure. Let's talk about tertiary structure. Tertiary structure is the shape of a protein molecule produced by regions of secondary structure. Secondary structure combined with overall bending and folding of the protein chain. So what's it doing now? So now I have the thing, it's an alpha helix, right? Boom, 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 boom. And I have another one, actually, that's going to go like this. So it's all the same, but now it's all wound up in this beautiful ball right there. See it? There. It's like two alpha helixes together, and they're all beautiful, and it's a really tightly knit ball because it's either a beta pleated sheet or it's an alpha helix, and it's like two of those. So it would be an alpha helix around this finger and an alpha helix around that finger, and it comes together to form. So that's why it's tertiary. So first it has the string, then it's the helix, then they come together for secondary structure. Now what is quaternary structure? Where could we possibly go from there? This is the overall structure of proteins composed of more than one polypeptide. So what did I do? I added another string of beads. So actually this is really cool. So let's say this is my giant polypeptide. Actually this is 3 polypeptides. So they're all lined up like this. This is what a peptide would look like. But if you look really closely, you can see it's actually 3 strands of beads. It's actually 3 polypeptides. So the primary structure is the one strand of beads, right? The one right there. The secondary structure is they're in a helix. The tertiary structure is they're in a helix connected to each other. And the quaternary structure is now there's the 3 strands have formed something and now they're one thing. Or you can think of it like this. Like here's our initial protein, right? See it? There's our initial protein. And then here's another protein. Here's another polypeptide. And now they've formed, and then here's our other one. So now all these guys together, we'll move it like this. What? I'm terrible at that. Now, look at that. Now we have a super beautiful protein right there. It's got 3 different strains and quaternary structure. Okay, let's move on a little bit. Okay. Oh, not that far. All the proteins in living organisms are built from, come on, can you guess? All the are built from the 20 amino acids. There's 20 of them. We will play with this in class. Each amino acid has a 3-letter code called a codon. 3-letter code. The 20 amino acids, only the identity of basically the side chain is what, because they all look the same. We just said that except for, well, we'll talk about it in just 1 second. I'll show you. Side chain. So for 20 of the amino acids, the only difference is the side chain. Then you have our friend proline, who's right here. Proline is a weirdo because it's got this like cyclical structure that comes back and binds with this nitrogen, which some of the rest of them don't do. So proline is a weirdo, but all the other 19, it's just the side chain, except for proline, who's a little bit of a problem. 2 nonpolar side chains are described as okay, so what do we have here? We have we are mixing them up into hydrophobic and hydrophilic. You already know the definition for these because you're super smart chemistry students. Hydrophilic. So we have hydrophobic, they hate water. They avoid aqueous solutions, nonpolar chains, gather into clusters to create a water-free environment. And then we have hydrophilic. So good news. Some of these little side chains and some of these things are hydrophobic, and some of the side chains are hydrophilic. So they will fold themselves that way so that you get, like these outside ones are probably, they love water, right? And these inside ones in here probably don't, and they fold themselves up like that. Your body is amazing. Okay. These 2 groups can undergo an intermolecular acid-base reaction to form, Zwitter ions. These are so cool. Please use the word Zwitter ion in a sentence. Zwitter ions. Okay, these are the coolest things. I love to use Zwitter ions just because it's so fascinating. So basically what happens is 2 groups undergo this intermolecular acid-base reaction and it forms a Zwitter ion. Just say it. It's so fun. Which is a neutral ion. It's neutral, but like half of it, this is like, this side is actually positive and the other side is negative, means the overall charge is neutral, but like there's a positive end and a negative end, and it's called the Zwitter ion. I just think it's so fun. 1 positive charge and 1 has a negative charge, thus they're neutral, but basically they're not neutral because one end, see, this nitrogen forms a positive end and this forms a negative end. This is threonine. And this is going to interact in all kinds of crazy ways, right? Because you've got a negative over here and a positive over here. So there's going to be some attractions and some bonds and some things that are happening because Zwitter ions are awesome. And they make all kinds of bridges and awesome stuff. Okay, next we're going to talk about amino acids. So I'll see you in a minute.